Assalamu alaikum wa rahmatullahi wa barakatuh. I am medical student and my aim is to provide them the best of the knowledge and the notes that help them even in their FSC and in the future MCAT inshallah. In the last video lecture, we have learned about the introduction of homeostasis, aspects of homeostasis, control system and feedback system. In the aspects of homeostasis, we have learned that there are three aspects of homeostasis: osmoregulation, excretion and thermoregulation. So today we are discussing osmoregulation in detail inshallah. What is osmoregulation? Maintenance of a balance between water and the solute content of the cell is called osmoregulation. Maintaining a balance, maintaining a regulation between the water and the solute content is called osmoregulation. How much the water must be inside the cell and how much it must be outside the cell, what will be the proportion of the solute content inside the cell and what will be its proportion when it is outside the cell. All this is regulated by the process of osmoregulation. Now, we must know that the water is a biological solvent. All the reactions, all the metabolic reactions that are taking place in our body are in the presence of water. Almost all the reactions take place in the presence of water. And even we know that most of our body composition is of water. So the water is a biological solvent and it moves by osmosis. Now what is osmosis? Movement of a water molecule from the area of higher concentration to the area of a lower concentration is called osmosis, or movement of a water molecule from the place where it is more to the place where it is less. In a one glass or in a one cell, we can say we have more water, then it moves to the cell where it has less water. So this movement is called as osmosis, and this movement does not require energy. Now what is water potential, pressure potential and solute potential? To know the osmoregulation, we must be familiar with these terms because we have to study separately osmoregulation in plants and osmoregulation in animals. So let us know something about these terms. What is water potential? Water potential is the potential of a water to move from one cell to another cell. Or you can say, take the example of these two containers. This container have a more proportion of water, have a more water molecule than this container, and this container has a more solute concentration than this one. So the water molecule in this container are more, so it moves from this container to this one because water molecules move from the area of higher concentration to the area of lower concentration. So water potential can be defined as ability of a living system to lose water. If we suppose that this container is a cell and this container is a cell too, so this cell loses the water and this cell gains the water, right? Because this cell has more water and this cell has less. So this cell loses the water. So ability of a living system to lose water is called water potential. Or you can say water potential is the concentration of a cell, and for the concentration, especially for the plant cell, we use the term water potential mostly for the plant cell concentration. And the concentration of the animal cell is expressed by the osmotic pressure. Now we have saw that in this container there are some more water particles and less solute, and in this we have more solute particle and less water. But suppose a container or a cell having no solute particles, only and only water particle or solvent particles, means that it has no impurities, then this water is called as pure water, and the water potential of the pure water is always 0. What is the water potential of the pure water? It is always 0. Now let's come to the solute potential. Now if we increase more and more solute to this container, its water potential decreases. What is water potential? Again remember, the more the water is, the more is the water potential, and the highest water potential is the potential of a pure water. So when we increase the solute particle in the container, the water potential start decreasing because we have more solute concentration. So we can say the lowering of the magnitude of a water potential is called solute potential. What is lowering of a magnitude of a water potential is called solute potential. So we can now say that this container has a less solute potential because it has more water, right? And this container has a more solute potential because it has more solute particle and less water particle. So now have you understand this definition? Lowering of the magnitude of a water potential. The more the water potential decreases, why water potential decreases? Because the amount of solute in the cell or in the container increases. Or here we have learned that the concentration of a cell, of a plant cell especially, is expressed as a water potential. And now the other thing, we know that the plant cell has a large vacuole, and this vacuole has mostly the concentration which is dilute. So the concentration of a cell sepse, what is cell sepse? Cell sepse is a vacuole. Concentration of a cell sepse is expressed as solute potential. Now we learned that more the solute enter into the container, the water potential start decreasing and decreasing and decreasing. So we can say that the water potential is always negative. Water potential is always negative, right? Now let's come to the next topic, which is pressure potential. Suppose that this is a container of a pure water, okay? It only has a water particles, and so the water potential of this container, what it must be? It must be 0. We have learned here that the water potential of a pure water is 0. When we applied pressure on the container, then what happened? That the pressure increases. Pressure of the cell increases, or pressure of the container increases. When the pressure increases, so we can say that the turgor pressure or the hydrostatic pressure of the container increases. So we can say that it increases the water potential. So we can know that the water potential is 0 of the pure water. When we add the solute particles, the water potential decreases and the solute potential increases. So we can write as the lowering of a magnitude of water potential is a solute potential, and solute potential is always negative. But when we applied the pressure, the molecules start moving so fastly and their kinetic energy increases, so the pressure potential is always positive. Or we can define it as pressure exerted by the cell sep of the cell or the cell solution. Means pressure exerted by even vacuole or the entire cytoplasm. It is called as pressure potential. So we know that when the water solute enters, water potential decreases, and when we increase the pressure, water potential increases. So we can write as water potential is lowered by the presence of solute and increased by water potential is lowered by the presence of solute and increased by the presence of the pressure. When we increase the solute particles in the cell or in the container, its water potential decreases because the number of the water particles become less and number of the solute particles become more. But when we increase the pressure, number of the solute particle increases. But when we increase the pressure exerted by the water particles on the cells, or the hydrostatic pressure produced by these particles, increases and the water potential increases. So now we can define it as water moves from the area of higher concentration from the area of higher water potential to the area of lower water potential. Water moves from higher water potential, this container has more water, so higher water potential, to the area of lower water potential. Or we can also say water moves from the area of lower solute potential to the area of higher solute potential. This container has a less solute particles, so less solute potential, and it has more solute particle, more solute potential. So move from less solute potential to the more solute potential. Water move from less solute potential to the more solute potential. Now let's come to our next topic, which is tonicity. What is tonicity? The strength of a solution in relationship to osmosis, or we can say it determines the direction of the movement of the water, from which cell to which cell water will move, and what will be the effect of the water potential in it. All these come under the topic of tonicity. In tonicity, we have three main terms: isotonic, hypertonic, and hypotonic. What is isotonic? It is same amount of water and solute. What is same amount of? Iso means same amount of water and solute. What is hypertonic? Hyper means more. What is hyper means? Hyper means more. So what is more? It is the more concentration of solute. So we can write as more solute. And what is hypotonic? Hypo means less. So lower solute or less solute. So what we have learned, that three main terms of tonicity: isotonic, hypertonic, and hypotonic. What is isotonic? The word itself says iso means same. Tonicity is the strength, means same water and solute outside and inside the cell. What is hypotonic? More solute. If it is used for the outside the cell, for the cell sac or the tissue fluid, so more content of solute in it. And if it is used for inside, so more content of solute is inside the cell. What is hypotonic? It is the less solute, whether inside or outside. It depends upon the statement what is given to us. Now, to understand these three main questions which are repeatedly asked in the papers, let us discuss first two main points. What are these? We have discussed earlier that the water moves from the higher potential to lower potential. What? Water moves from higher potential to lower. Let's come to our next topic, which is tonicity. What is tonicity? The strength of a solution in relationship to osmosis, or we can say it determines the direction of the movement of the water, from which cell to which cell water will move, and what will be the effect of the water potential in it. All these come under the topic of tonicity. In tonicity, we have three main terms: isotonic, hypertonic, and hypotonic. What is isotonic? It is same amount of water and solute. What is same amount of? Iso means same amount of water and solute. What is hypertonic? Hyper means more. What is hyper means? Hyper means more. So what is more? It is the more concentration of solute. So we can write as more solute. And what is hypotonic? Hypo means less. So lower solute or less solute. So what we have learned, that three main terms of tonicity: isotonic, hypertonic and hypotonic. What is isotonic? The word itself says iso means same, tonicity is the strength, means same water and solute outside and inside the cell. What is hypotonic? More solute. If it is used for the outside the cell, for the cell shape or the tissue fluid, so more content of solute in it. And if it is used for inside, so more content of solute is inside the cell. What is hypotonic? It is the less solute, whether inside or outside. It depends upon the statement what is given to us. Now, to understand these three main questions which are repeatedly asked in the papers, let us discuss first two main points. What are these? We have discussed earlier that the water moves from the higher potential to lower potential. What? Water moves from higher potential to lower. So what it is? Water moves from higher potential to lower potential. Now the next thing is that water moves from hypotonic solution to the hypertonic solution. What? Hypertonic movement take place from hypotonic hypotonic to hypertonic. What we learn about hypotonic? That lower concentration of solute. Lower concentration of solute means more water potential. These are two same thing, but in the different words, because in different books it is written in the different forms. So water moves from hypotonic to hypertonic, from lower concentration of solute to the higher concentration of lower concentration of solute to the higher concentration of solute. So now let's see what is the first question. What happens when a plant cell is placed in a pure water? What is pure water? Pure water is the solution having no solute, means it is hypotonic. Lower concentration of solute, it is minimum, less or zero. So our external temperature is hypotonic. Okay. So when a plant cell is placed in the hypotonic solution, what happens? And inside, of course, when the outside is hypotonic and the inside is hypertonic. So water moves from hypotonic to hypertonic. That means water moves in this case from the outside the cell to inside the cell, or we can say endo-osmosis occur in this case. In the first case, we can say water moves from outside to inside the cell. Water moves from, what has happened? Water moves from the area of a lower concentration of solute to the area of the higher concentration of solute. Water moves from hypotonic to the hypertonic. Plant cell must have the solute in it. We know that there are many minerals, vitamins along with the water in the cell and many other cell organelles. So it is not the pure water at all. But when it is placed, the cell is placed in the pure water, what happens? That the water moves from outside the cell to the inside the cell, moves from outside to inside, and what it is called endo-osmosis. Endo means inside the cell. Now let's see the next question. Why the plant cell not burst by endo-osmosis and animal cell burst? Why? Because the plant cell has cell wall. And the cell wall maintains the turgor pressure, it resists the turgor pressure, it resists the internal hydrostatic pressure. Plant cell has cell wall, and we all know that the animal cell does not have the cell wall, so it does not resist any internal temperature. When more and more water comes inside the cell, in the animal cell, what happens? It eventually bursts out. But what happens in the plant cell? When more and more water comes inside the cell, internal hydrostatic pressure increases and it resists the tension and it stops the further inflow of water inside the cell. Okay. Plant cell has a cell wall, while animal cell does not have cell wall. It's so simple. Animal cell has no cell wall. What happens when the plant and the animal cell plays in the hypertonic solution? Means the cell itself is hypotonic and it plays in the container which has more and more solute. It is hypertonic. Hypertonic is more solute. So water moves from hypotonic to hypertonic, or from the area of a higher concentration to the area of a lower concentration. So what happens in this case? Exoosmosis occur. Exoosmosis occur. When the exoosmosis occur, what happens? That the plant cells become flaccid and animal cell crinkled up like the crenation of the RBCs. Now, when the endosmosis occur, plant cells maintain the temperature. Plant cell maintains the internal hydrostatic pressure. But what happens? How can the animal cell compensate with the situation? Animal cell is mostly like our body tissue fluid, and our cell has the same tonicity. It is like our cells plays in the isotonic environment, so that there is no movement from outside to inside the cell of the water particles, most probably. Okay, so for today it is up, and thank you so much for listening. Hope you like my video. If you have any queries regarding any topic, you are always welcome in the comment section, or you can DM me on the Instagram Study with Doctors. Don't forget to subscribe my channel so you get notified when my next video is uploaded. Assalamualaikum warahmatullahi wabarakatuh.